In this video, we'll be looking at cell transport. So transport proteins allow the passage of hydrophilic substances across the membrane. Some transport proteins, called channel proteins, have a hydrophilic channel that certain molecules or ions can use as a tunnel. Channel proteins, called aquaporins, facilitate the passage of water. So water passage, or osmosis, happens through aquaporins. Other transport proteins, called carrier proteins, bind to molecules and change shape to shuttle them across the membrane. A transport protein is specific to the substance it moves, so aquaporins only move water. They're not able to move things like glucose or ions or anything else. Diffusion is the tendency for molecules to spread out evenly in the available space. Although each molecule moves randomly, diffusion of a population of molecules may exhibit a net movement in one direction. At dynamic equilibrium, as many molecules cross one way as cross the other. So here's an example of dynamic equilibrium. So although these molecules here may continue moving, there will be no net movement. Whereas back here, because the molecules of dye were predominantly on the one side of the container of water, and they are going across a membrane in which the dye molecules are small enough to cross, eventually most of the molecules had a net diffusion, in this case, across to this other side until they reached equilibrium. So it's dynamic equilibrium because the molecules can still continue to move. Passive transport. Substances diffuse down their concentration gradient, the difference in concentration of a substance from one area to another. No work must be done to move substances down on the concentration gradient. The diffusion of a substance across biological membranes is passive transport because it requires no energy from the cell to make it happen. So here's net diffusion of the orange particles, and that just like we've seen, but now we've added in a separate solute, a purple solute. So this purple solute is also able to pass through the selectively permeable membrane. So it passes through as well, so both diffuse to dynamic equilibrium, but they are diffusing, net diffusion is happening in different directions. Osmosis is the diffusion of water across a selectively permeable membrane. Water diffuses across the membrane from the region of lower solute concentration to a region of higher solute concentration. So this is different from the way that we think of osmosis. We're not talking about the movement of water sometimes itself. We have to first look at the solute concentrations, because it's not always possible for the solutes to move from one area to another because of selectively permeable membranes, but water can usually travel through those membranes, and so that will sort of equalize what's going on. So here's an example. In this case, we have a lower concentration of solute on the left side and a higher concentration of solute on the right side. And we have a selectively permeable membrane, which will allow water to move across it, but won't allow sugar to move across it. So it's selectively permeable for water, allowing it, but not sugar. So what we see actually happening in the process of osmosis is that molecules of water on this left side with a lower solute concentration are free. So they are able to, they're not bound to any of the solute, but on the right side, we see many of the molecules are bound or attracted to the solute. So that means that there's less free water. So these molecules of water that don't have any attachment to anything. So in that way, even though it looks like there's water on both sides, there's more free water on the left side. So over time, some of that free water is going to move to the right side and increase the amount of water on that side and decrease the solute concentration. All right, tonicity is the ability of a solution to cause a cell to gain or lose water. Isotonic solution is a solution concentration that is the same as inside the cell. So there's no net water movement across the plasma membrane. In a hypertonic solution, the solute concentration is greater than inside the cell, so the cell will lose water. And in a hypotonic solution, the solute concentration is less than inside the cell, so the cell will gain water. So for isotonic, hypertonic, and hypotonic solutions, we're always describing it in relation to something. You can't just say a cell is hypotonic. You have to say it's hypotonic to something else. It's the same way that you can't say so-and-so is taller than. If you're talking about a student being taller, you have to say they're taller than someone else. All right, so here's an examples of solutions in hypertonic, hypotonic, and isotonic solutions. So first of all, if we have an isotonic solution, this means that there's no net movement of water into or out of an animal cell. In the case of a plant cell, the same thing is happening. There's no net movement into or out of the cell. For an animal cell, this is an ideal environment. We want isotonic solutions for animal cells. For a plant cell, if we have no net movement of water, that's actually going to make the cell flaccid or wilting. It's not going to be the ideal environment for plant cells. Plant cells' ideal environment is actually a hypotonic solution. So a more watery environment with less solute is going to cause a constant inflow of water into the cells. And this is part of how plant cells maintain their turgidity, which is their ability to be strong and upright, is due to the finding lots of water and being in the hypotonic solution. On the other hand, if you put a red blood cell in a hypotonic solution, so a watery environment, you're going to cause the cell to swell and potentially even burst. And this because there's a constant inflow of water and there's no way to get rid of that. Okay, so the bell rang on me and that stops what I was saying. So back to a hypertonic solution. This is the last solution. When you have a hypertonic solution, it causes a net movement of water out of the cell in both plant cells and animal cells. And so when water moves out of the cell, they will become shriveled up and the plants will become plasmolized. So that is for a hypertonic solution. So those are the three types of solutions. Facilitated diffusion. In facilitated diffusion, transport proteins speed the passive movement of molecules across the plasma membrane. Channel proteins provide corridors that allow specific molecule or ion to cross the membrane. Channel proteins include aquaporins, which you've seen already. Aquaporins facilitate the diffusion of water or osmosis. And ion channels that open and close in response to stimulus are called gated channels. Carrier proteins undergo a subtle change in shape that translocates the solute binding site across the membrane. So when we say that, we're saying that the carrier proteins are going to have a change in shape, is actually going to change them, move the overall protein, and that change in shape is going to cause the solute binding site to change from one area to the other. So here's an example of a channel protein and a carrier protein. So in the channel protein on the top, here's the movement of solute. So you can see the solute is moving from an area of high concentration down to an area of low concentration, but it can't go directly across the plasma membrane because it is too large or has a charge. So instead it has to go through a channel protein. So a channel protein is just gated. It's open, but only for this particular molecule. In a carrier protein, it's similar, but it's a little bit more complex because the carrier protein actually changes in shape in order to move the solute across. So again, we're going from an area of high concentration to an area of low concentration. But in this case, we are looking at the solute binding to an active site of the protein, and that binding site then causing the solute to move across. And so this is still facilitated diffusion. It's still passive transport. There's no energy required because the cell is just is not required to spend any of its own energy to make this happen. Okay, then we get to active transport. So active transport is different because it uses energy to move solutes against their gradient. Facilitated diffusion is still passive because the solutes move down its concentration gradient, but for some transport proteins, have to move solutes against their concentration gradient. So active transport moves substances against their concentration gradient, or from low concentration toward high concentration. So it's opposite of the way we would expect them to move. Active transport requires energy, usually in the form of ATP. Active transport is performed by specific proteins embedded in the membranes. So the first one we're going to look at is the sodium-potassium pump. Artificial active transport allows the cell to maintain concentration gradients that differ from their surroundings. Sodium-potassium pump is one type of active transport system. Sodium-potassium pump is found in animal cells. So here we see the cytoplasmic sodium binds to the sodium-potassium pump. So this is inside the cell. We have a binding of 3 sodium ions to this protein. And then that is going to cause for conformational change. That's going to cause for binding of the ATP to the sodium potassium pump. And that binding is going to cause a conformational change. So we see the conformational change, it changes to go in the opposite direction. So now we have 3 sodium ions that are outside the cell in the extracellular fluid. And the sodium no longer binds because of the change of shape. But this now is the correct conformation for potassium to bind. So potassium is going to come and bind. When potassium binds, it's going to trigger the release of a phosphate group. So the phosphate group is going to leave. And the loss of the phosphate is going to cause the protein to go back to its original shape. So it's going to go back to facing inward. And then the 2 potassiums are going to leave. Now the cycle can repeat. Now it's open so that sodium can bind here, and the cycle can start again. So let's just go through that one more time. So 3 sodiums enter here. The ATP donates the phosphate. 3 sodiums leave the phosphate group. For 2 potassiums bind, the phosphate leaves, change in shape, the two potassiums leave, and now we're back to our beginning. Okay, so there it is, 1, 2, 3, 4, 5, 6 as a cycle. Okay, so just as a review, this diagram is nice because it shows all the types of transport together. So we have diffusion, simple diffusion, straight through the membrane. This would be things like carbon dioxide. We have facilitated diffusion, which is still passive transport, and it either goes through a channel protein or a carrier protein, but it doesn't require any energy from the cell. And then we have active transport, so where we're working against the concentration gradient. So in this case, we're moving these 3 sodiums out of the cell, even though at this point there are more sodiums outside the cell than there are in. And we're moving the potassiums inside the cell, even though there are fewer potassiums outside in the extracellular matrix than there are inside. And so this always requires ATP. So if you ever see concentration gradient movement of materials against a concentration gradient, you can expect that ATP has to be involved or some other form of energy, but generally ATP is involved. Okay, cotransport. Coupled transport by a membrane protein. An electrogenic pump is a transport protein that generates voltage across the membrane. The sodium-potassium pump is the major electrogenic pump of animal cells. The main electrogenic pump of plants, fungi, and bacteria is the proton pump. Cotransport occurs when the active transport of a solute indirectly drives the transport of another solute. So plants commonly use the gradient of hydrogen ions generated by proton pumps to drive active transport of nutrients into the cell. So there's the proton pump. We just have the movement of protons against their concentration gradient from here out into the extracellular fluid. And then we use this proton pump. So we're moving protons against the gradient, and then we're allowing the protons to come back through, which they can't come back through the phospholipid bilayer because they're charged. But we're using that with a sucrose proton cotransporter. So that cotransporter is going to move sucrose into the cell, and sucrose we want in a higher concentration inside the cell than outside. Okay, so that is the cotransport. Now we're looking at bulk transport. So bulk transport across the plasma membrane occurs in 2 ways, exocytosis and endocytosis. So small molecules in water enter or leave the cell through the lipid bilayer by transport proteins. But large molecules such as polysaccharides and proteins have to cross the membrane in bulk via vesicles. So bulk transport requires energy, so it is a form of active transport. Exocytosis is transport vesicles migrate to the membrane, fuse with it, and release their contents. So exocytosis is the movement of materials out of the cell into the extracellular space. Many secretory cells use exocytosis to export their products. So secretory cells that are creating materials like hormones or other glands would use exocytosis. Endocytosis, the cell takes in macromolecules by forming vesicles from the plasma membrane. So endocytosis is a reversal of exocytosis involving different proteins. There are 3 different types of endocytosis. Phagocytosis, or cellular eating. A cell engulfs a particle in a vacuole, and the vacuole fuses with the lysosome to digest the particle. Penocytosis, cellular drinking. Molecules are taken up when extracellular fluid is gulped into tiny vesicles. So in this case, we're just taking up the fluid that's surrounding the cell. And receptor-mediated endocytosis. In receptor-mediated endocytosis, the binding of ligands, a molecule that binds specifically to the receptor site of another molecule to the receptors, triggers vesicle formation. So we don't have receptor-mediated endocytosis is more complex and happens more rarely because, well, not more rarely. It's just more complex because you have to have the binding of a specific molecule to the receptor site in order to trigger it. Okay, so here are the 3 types. Oh, I thought I would have zoomed in on that. Sorry about that. So phagocytosis, penocytosis, and receptor-mediated endocytosis. So for the extracellular fluid taking in something for phagocytosis, that's like a food particle or possibly something like a bacteria or something. It's has the cytoplasm forms around, it spreads out around it, and the plasma membrane comes around, and then it is engulfed into the cell. In penocytosis, it's just the dissolved solutes. And then in receptor-mediated endocytosis, you have receptors on the surface of the membrane. And when those surface membranes bind to the product that we're looking for, that launches into the formation of the vesicle. And so the vesicle, the membrane encloses around it, and then you have a membrane-filled vesicle. And we call it a coated vesicle because